Good morning, everyone. It is my great pleasure to welcome everyone while still some people join our meeting. My name is Oliver Fien, Director of the West Coast Metabolomics Center. It is my great pleasure to welcome today this month's seminar speaker, Dr. Maria Fedorova. She studied biochemistry at St. Petersburg State University in Russia, and then obtained her PhD at the Faculty of Chemistry and Mineralogy at Leipzig University in Germany. She worked as a group leader at the Institute of Bioanalytical Chemistry, Faculty of Chemistry and Mineralogy at the University of Leipzig. And in August 2021, Maria's group moved to the Center for Membrane Biochemistry and Lipid Research at the Technical University of Dresden in Germany. Her research is focused on development and implementation of lipidomics and bioinformatics solutions to address complexity and plasticity of lipid metabolism in a variety of biological systems. In particular, Dr. Fedorova aims for deeper understanding of pathophysiology of metabolic and inflammatory disorders, including obesity, insulin resistance, type 2 diabetes, and cardiovascular disorders. Please help me welcome Dr. Maria Fedorova from the University Hospital and Faculty of Medicine at the Carl Gustav Karas of Technical University, Dresden. Maria.
Thank you very much. So I guess I will share my screen now. So thank you very much for inviting me to talk today at this seminar. And so I will be talking today about our bioinformatics solutions to support throughput and accuracy of lipidomics data and processing. And the first thing which I'd like to mention that I totally would like to co-author this presentation with Jishu Ni, who is a postdoc in my group. And he's actually the one who is making all our bioinformatics dreams true and making it from the idea actually to the product. And as Oliver just mentioned, so we just moved from University of Leipzig, where we were for last almost 10 years, I guess, to the Technical University of Dresden. So that's a new affiliation, but the most of the work which I will show today is, of course, was done back in Leipzig. So before I start with our bioinformatics solution, just to mention you what we actually doing very generally. So our research group is called Lipid Metabolism, Analysis and Integration. And we are interested about lipids and lipidomes on all different levels. So we look in the single lipids, in the lipid collectors, but also in the lipidome as their biological system in question. And in this respect, we are particularly looking for the lipid metabolism and stress responses, so lipidome plasticity and remodeling, as well as lipid and lipidome quality controls in different levels. In terms of signaling, and we very much interested in the concept of epilipidome, and I will talk about that later on a little bit. So this is usually modified lipids, subset of natural lipidome, which is formed by lipid modification, so aiming to increase regulatory capacity of biological systems. And in terms of biomedical focus, so we look into metabolic diseases, mostly obesity, diabetes, liver diseases, inflammatory disorders, and looking for possibilities of pharmacological intervention via targeting lipid metabolism, and also using lipids as a possible biomarkers in terms of mechanistic, maybe predictive or diagnostic purposes. So that is quite a broad range, of course, and we put lipidomics here in center to address, for instance, reference lipidomes, for instance, of adipose tissue, that give us access to the possibility to translate our finding to the clinical applications, again, in terms of obesity, maybe cardiovascular diseases, but also looking in the lipidomes in details, we can figure out regulation of metabolic pathways, which could give some mechanistic understanding of the problem, leading to the possibility of pharmacological targeting. So in our group, we do a lot of analytics, so mass spectrometry-based lipidomics. And I will not gonna talk today a lot about that, so the methods which we use for that, but in this respect, we also focus a lot into the different types of sample preparation, but also into the chromatography and mass spectrometry solution, for instance, optimal mobile phases or stationary phases for the chromatography, in-source fragmentation effects, optimization of Msms methodologies, and solution for the quantifications. So once again, I'm not focusing in this presentation about these analytical questions which we are addressing in the lab as well. But if you're interested, I will advise you to have a look on the presentation or in the publication, which is available on the bioarchive, but it's also coming out, I guess, in a few days. Here we used, so we created a reference lipidome of human white adipose tissue, where we applied all this methodology available in our laboratory to these purposes. So different types of chromatography, different types of mass spectrometry, retention time mapping, lipid identification, and so on. And I would like to mention Mike Lange, who is now actually not far from you at the University of California in Berkeley, who was the leading person in this Adipo Atlas development. So if you're ever interested in doing a very deep lipidomic scan, so you know where to find him. So once again, in this talk, I would rather focus on the analytical solutions. So if we have our nicely optimized method for the sample preparation for lipid fractionation, liquid chromatography optimization, and mass spectrometry, and if everything of that works perfectly fine, we are actually coming to the point when we collect a lot of lipidomics data. And in this respect, when we started back, maybe 8 years ago, looking into the lipids first, we faced this big problem with a lipid identification. I must say, I did my PhD and my postdoc in the proteomics. And when I started to do lipid as my independent research, maybe one of the most shocking experience was that after you optimize and can measure your analytes, you actually have almost back then no tools to proceed with the high throughput identifications. So in the beginning, of course, to understand how all that works, we did a lot of manual lipid identification. And in this case, of course, we would follow pretty much this standard scheme. Yeah, so we look at the chromatographic peak, we get our signal, get extracted chromatograms. With a high resolution instrument, we can determine elemental composition, propose a bulk structure of the lipid, and go back to the MS2 spectrum, where we can look at the corresponding fragments and identify what kind of lipid it is. So following this way, and after optimization, some LCMS parameters, so a regular Msms acquisition with data-dependent acquisition run over 30 minutes give you a few thousand of MS2 spectra. So the manual analysis was at certain point, of course, out of question, and we had to move to the computationally assisted data processing, where we can process our data sets in the reasonable time frames. So, and here Ni decided to step in and actually moved from the wet lab application into the programming, and developed the first tool which we have in our lab is Lipid Hunter. So Lipid Hunter is a software which dedicated to identification. First, it was done for phospholipids from LCMS and shotgun lipidomics datasets. So what a Lipid Hunter would do, it would do exactly the same like you would identify your lipids manually, and it will look exactly at the same parameters, but it will do just it's much faster than a person can do. So the idea behind the whole workflow in the Lipid Hunter is to get extracted ion chromatogram, to get corresponding MS1, to identify elemental composition, to propose certain possible structures, and then to look into the corresponding Msms spectrum and to match different ions to the certain lipid compositions. Since then, many things changed. So now we have a version which is called Lipid Hunter 2, which is covering just more lipids. We have phospholipids and lysophospholipids. We have glycerolipids and also optimize it for different types of sphingolipids as well. And Georgia also participated in this optimization workflow here as well. So Lipid Hunter is totally open access, so everybody can download and use it. And the idea behind that it's not working with any spectral libraries, either experimental or in silica created, but it's based on the rule-based identification approach, which we call here a bottom-up approach. So there are several different features in Lipid Hunter, and the core of that is actually identification of different fragmentation mechanisms. So for all different lipid classes, we have created by looking into many MS2 spectra, this kind of fragmentation pathways where we know the rules and how each lipid will be fragmented. So we would implement these fragmentation rules and provide to Lipid Hunter as a white list what kind of fatty acids, what kind of head groups, what kind of sphingosine bases we can have in our lipids, which is also very flexible. And it will use those lists to match the fragment according to those fragmentation. So there are different scoring algorithms involved in Lipid Hunter. The first one is isotope score for the accurate assignment of elemental composition. So I guess all of you know here that there is a lot of isotopic overlaps. And it's important to define a really correct isotopic patterns so that we are not using M-1 or M+1 isotope as instead of monoisotopic signal. The final scoring algorithm in the Lipid Hunter is called rank scoring for bottom-up identification. And here we decided not to use any kind of intensity measure, but rather allow Lipid Hunter to rank the present fragments by their intensity. And this rank index will be used to calculate the fragmentation rank. And then together with the weight factors, which again are user defined, that would give us the total score for the lipid identification. Why is that nice? Because first of all, we're independent on the absolute intensities, and even in the mixed MS2 spectra can identify the species pretty accurately. And providing this kind of weight factor by the user, you can easily optimize to your collision energy which you are using in your instrument or any other specific settings. Yeah? So the fragmentation mechanism, not mechanism, but at least yields might differ between different vendors and different setups of your mass spectrometry experiment. So then we also made here some visualization options because we still, of course, we very much rely on our Lipid Hunter, but still we would like to see how good is our identification afterwards. So together with the Excel report, you are also where you have all the parameters and provided. You can also get this HTML-based page where you can click on each identification and you are getting these 6 panels where you have, or at least for me, for instance, as a user, there is a lot of information which I can very easily visually control. For instance, I could see here the extracted ion chromatograms for this particular mass to charge ratio, and also I can see where exactly Msms was taken. And of course, now it got better, but in the previous generation of the instruments, it was not always taken on the apex of the peak. So then we would know that we have also optimized a bit our acquisition methods as well. So we can see here, for instance, it's taken pretty much on the apex of the peak. We can see here MS1 scan corresponding to that time point here, isotopic score distribution to confirm that it's actually rightly assigned monoisotopic mass. We can also see here the full MS2 spectra and then the zooms in the MS2 spectra where these fragments which were actually used for the identification would be marked. And here we could see in green or in blue the fragments which are correctly assigned or fitting our identification, and in red the ones which actually might correspond to something else. And of course, there are cases where we can judge by the presence of this red signals if the identification was not entirely correct. So once again, now we have this version of Lipid Hunter 2 as an open source software. So it's available on our GitHub page, and it's also available with a GUI interface and working on all different platforms. And we also provide here, for instance, integration option into the 9 workflows, so people who are using 9 might be interested in that as well. So, and Lipid Hunter, of course, works for normal unmodified lipids. I mentioned to you before that one of the other big interest in my group is study of modified lipids or lipid modifications in general. And just before moving to the next tool, I would like to say a few words. Why do we think that's important? So we all know that lipids are very, very diverse. We have different head groups, we have different backbones, we have the whole range of different fatty acid chains. And all these combinations form this beautiful diversity of lipidomes. But lipids also undergo different modifications. So we are introduction of small chemical groups, and that can happen enzymatically or non-enzymatically in different ways. To give you a few examples, let's have a look on this PAPE lipid, and it can get oxidized, for instance, via the action of lipoxygenases, or even through the free radical driven hydroxylation, and that can lead to the formation of lipid hydroperoxide. So this lipid has already changed physical chemical properties and actually have another different functional outcomes. Another example is a modification on the head groups. For instance, here in ethanolamine lipids, this amino group can modify by the reducing sugars such as glucose, and that can happen, for instance, in our bloodstream in the case of diabetes when the glucose concentration is pretty high. So this is called non-enzymatic glycation or Maillard reaction, which would lead to the formation of glycated PAP. So that molecule can be used, for instance, as a biomarker of the diabetes. And if you know about hemoglobin, glycated hemoglobin, actually exactly the same reaction between amino group and reducing sugar glucose is already a well-established biomarker which is used in the clinical diagnostics. So a different modification can happen in different way. And we formulated here a concept of epilipidome as a subset of natural lipidome formed by lipid modification via enzymatic, non-enzymatic reaction, variety of them required to regulate complex biological functions. So it's something very similar in the idea of, for instance, if we talk about DNA methylation, yeah, this is an important factor in epigenomic regulation. Of course, everybody knows about protein post-translational modification, phosphorylation, acetylation, many other of those one. So again, introduction of small chemical groups and amino acid residues in proteins. So this proteoforms, of course, already very well acknowledged signaling molecules, yeah, in different signaling pathways. So in this way, lipid post-translational modification, if we call it this way, forming this epilipidome again with a gain of regulatory capacity in the biological systems. A few examples, some of them you for sure know or maybe didn't think about in this perspective is, of course, inflammation and its resolution, yeah, with the formation of oxylipins. So arachidonic acid, when it's released from the phospholipids, can get modified under the action of COX-2 enzyme and some other prostaglandin synthesis, for instance, with the formation of prostaglandin E2 or many others. And there are specific receptors, and their expression is tissue dependent, which can afterwards, through the signaling, leads to the activation of macrophages, for instance. Yeah? And each of you in your life, at least once, I guess, inhibited this reaction by taking aspirin or ibuprofen. The other, maybe a less known example, but something that becomes very popular nowadays, is a so-called therapeutic cell death. And this is a new type or newly described cell type, cell death type via necrosis, where cell dies due to the massive lipid peroxidation, and for instance, certain classes of phospholipids are supposedly involved in the mediation of this kind of cell death via oxidation mechanisms. And of course, in the area of innate immune responses, and especially in the chronic inflammation, which is accompanying atherosclerosis, oxidized lipids are known already for a few decades, where they can form so-called damage-associated molecular patterns exposed, for instance, on the surface of the cells or on the surface of the lipoproteins, and they can be recognized by the variety of pattern recognition receptors such as RAGE receptors, CD36, or toll-like receptors as well. And in this case, there would be not only oxidized phospholipids, but also oxidized cholesterol esters, and what is relatively less studied is oxidized fat cell glycerols. So we are interested in looking into this mechanism, what happens in terms of lipid modifications in the biological systems. And of course, we use mass spectrometry to do that, and also optimize a lot of settings, parameters to have our measurements successful. But at the end of the day, we again face the problem of identification. So we cannot always go manually here, and we want to have a tool which would allow us successfully identify modified lipids as well. So the problem with modified lipids is actually absolutely a numerous search space. So if we know that there are so many different unmodified lipids, so the modifications, even if we're talking about just oxidation of lipids, are increasing this diversity or potentially increasing this diversity by many, many fold. Yeah? So this is a complicated scheme. I'll just want to illustrate here that we have a variety of different modifications, for instance, hydroperoxide, hydroxide, keto group, epoxide, truncation of the chain. If I have a fatty acid with more than 3 double bonds, we have formation of these prostaglandin rings, and diversity goes high and high. So once we calculated here that what can be estimated diversity of oxidized lipids or this epilipidome, and you can see this number, so this is absolutely crazy. And we don't know what is that true or what is that actually biologically possible. So this enormous search space, of course, making our identifications or finding the tool or solutions how to identify oxidized lipids very challenging. So here we came up with another software. So Nick came up and Georgia helped him as well, which we call Lpp Tiger. So Lpp stands for lipid peroxidation products, and Lpp Tiger software has 3 interesting algorithms. So of course, this is software for identification, yeah, the sort algorithm targeting that. But also Lpp Tiger can predict oxidized lipids, and we also developed different fragmentation algorithms for that. So in terms of in silico prediction of oxidized lipidome, we didn't go through the simple enumeration of oxygen in all different positions. So we went actually through the other way. So we read a lot of papers about fatty acid and lipid oxidations and the chemistry of those reactions in vivo and in vitro, and reconstructed oxidation metabolic networks for 10 different polyunsaturated fatty acids. So you see here example, I think, for linoleic acid. So all the reactions which can lead to the formation of different oxidation products. And we did it for all 10 PUFAs. And you are not meant, of course, to understand this in details, but just to show you that it's quite a complex network where we have more than 480 lipids or oxidized fatty acids characterized by more than 500 reactions. And that is considering enzymatic, but also non-enzymatic reactions as well. So then Lpp Tiger is actually using these networks and rules defined from these networks to perform this in silico prediction of epilipidome by using these units or blocks of the polyunsaturated fatty acid, which you give in as input in order to perform all this kind of oxidation reactions, and giving you a sample specific, or your input specific and silico predicted epilipidome. and you are getting it in a form of such as the F files, which you can actually use for many different purposes, and we actually do it nowadays this way. So the next point was in silica fragmentation. So we wanted to predict how oxidized lipids fragment to have the library of their MS2 spectrum from this oxidized in silico epilipidome. And here we went for developing the fragmentation rules. And of course, oxidized lipids, they do not fragment exactly the same like unmodified lipids. So we had to do quite many optimizations in this way as well. So there are not much standards which you can use for these purposes. And here we had to prepare a lot of in vitro oxidized phospholipids, neutral lipids mixtures, separate them accurately by chromatography to resolve isomers, and then fragment them and actually look very carefully into their structures, into the fragments to define different structures. So that gave us all those fragmentation patterns and rules, and Paulina and Angela also made a great job in putting that all together, because this is quite complex process. So those rules again were implemented into Lpp Tiger, and then so the next step after the prediction of oxidized lipidome for Lpp Tiger is to create the MSP library of in silica fragmented oxidized lipids, which were predicted. So the next logical step would be to match our libraries with experimental data. So, and this is one of the scoring algorithm implemented in Lipid Tiger. So we wanted to be here super accurate because, yeah, those oxidized lipids are very tricky ones. So we implemented here 5 different scores, and one of those is spectral similarity score, which is based on the matching of our experimental data with the ones derived by in silico fragmentation. We also have our rank score, which we took from a Lipid Hunter algorithm, isotopic score as well. And here we very nicely implemented 2 other scores, which allows to access the specificity of your identification, and also maybe to provide you even more structural details on the position of modification, for instance, as well. So that is all summed up in so-called overall score, which you are getting at the end of your identification process. So again, here we have the similar 6 panel identification output. And we have a pretty similar idea here. So extract the time chromatogram, MS1, then the topic score, and here this mixed panel with different spectral similarity matching, and so on and so on. Yeah, so the Lpp Tiger is again absolutely open source software and available at our GitHub. It has a similar user interface as Lipid Hunter, so everybody can use that. And if you have any question, you are more than welcome just to contact us and we can help you with it. So now we are working on the new version of Lpp Tiger, Lpp Tiger 2.0, where we want to introduce more modification position-specific fragments, and also to use retention time method, which is for which we defined also the elution order rules for different isomers. And the current version of Lpp Tiger is working with phospholipids, but now we are enlarging it also for the neutral lipids, because in our experiments we actually see a high significance of those lipids as well. Another very nice feature here that we collaborate very closely with LipaStar developers, or scientific part of the team of the LipaStar software, and this is a very nice software for high throughput identification of lipids. And we actually team up here that LipaStar can predict potential oxidized lipids, and Ani made a lot of work to build this interface here together with LipaStar developer, that those theoretically possible oxidized lipids from LipaStar, or potentially possible, those MS2 spectrum will be actually directly translated into Lpp Tiger, which will perform this more specific identification, and then actually will feed back the results to the LipaStar version 2. So if you are using LipaStar, so you can also try to and look into this implementation, which of course speed up the whole process pretty much. Okay, so now I describe you two applications for our data analysis part. So where we have identification software directed for the unmodified lipids and also for lipidomes. So we actually never focused on the software solutions for lipid quantification because there are a lot of tools out there, either commercially available or actually free, which allows you to perform quantification from LCMS data. So because of course with some specificity, but chromatographic MS peak is pretty much similar between different kinds of biomolecules in some respects. So the other solutions which we have here, it's some kind of solutions for visualization. I will not focus much, but just show you one example of a tool which we call Lipid Circles. And that allows you to build different kinds of relationships between types of lipids and also express it in different manner. Here is an example, for instance, for mapping oxidized lipids. And we can display here the precursors or the parent lipids from which those oxidized lipids can come from. And then we can have a distribution of different oxidized fatty acids here. So for instance, linoleic acid here, we see that hydroperoxides were really the most identified modification in this particular fatty acids. And this fatty acid was actually coming from phospholipids a lot, and also, for instance, from CG and so on. And this lipid circles, we don't have a graphical user interface, but we can use it in different other ways, but it can build different types of relationship for different lipids here. So the next solution after we actually successfully can measure, identify, and even visualize our data, the logical way would be to go to the integration of our lipidomics results. So where we want to look into different networks, pathways, analysis, so actually to make a biological sense out of our data. And actually when we wanted to move in this direction as the next logical step, we figured out that there is quite a gap which is not filled. And for instance, if we want to look in our different lipids from different data sets, and maybe we want to combine it in with multi-omics data sets, it's not so easy and straightforward to do just because of the nomenclature, notation, and different ID issues. Yeah? So if we want to go to the network analysis, which of course still very difficult for lipids for many other reasons, which I'm not mentioning today, we have to actually spot different identifiers. For instance, CABI is one of the standard ways which can be translated in the different genome scale networks which are available out there. But how we can support this high throughput translation or of this conversion of the names? And if you look in this issue actually in more details, there are much more problems coming up, and I will show you some examples. So, and in order to show this gap, we actually developed here another tool which we called Lipid Links. So one of the biggest problem, maybe not obvious, but still very tedious in terms of integrating large lipidomics data sets, especially if they are derived from different software tools, for instance, your own data set and the data sets from the publications or databases or repositories on the same biological systems, for instance. So they all can have a very different way on the notation. Yeah, so like we all speak different languages, and the same way people are used to write lipids in a very many different ways. Yeah, you see here the structure of one lipid, and you could see just an example in which this lipid can be, in which way this lipid can be written. So if we want to integrate lipidomics data, a big lipidomics data set, it's obvious we cannot spend time by looking at every single lipid in a list of hundreds of lipids to define which name is actually correct and which one we should take. So for that purpose, we developed Lipid Links, which is a data transfer hub, as we told. So the main idea here, so we looked through the different lipids classes and so on. So mostly mammalian lipids. We didn't cover any bacterial or plant lipids here. And for the different notation systems. And we wanted, so we developed here a converter and the equalizer modules, which allows to convert all those different 17 identifiers, or it's also flexible, you can put more in any particular output you like. Yeah, so we can convert those all different names in the shorthand notations, in the CompDB, for instance, from Lipid Maps system, and we have our own Lipid Links, what we say, computationally friendly solution here. And then we also have this equalizer models because it's also important actually to bring your lipid to exactly the same level of annotations if you want to compare different data sets. Yeah, for instance, somebody reported bulk composition, total carbon number, and double bonds, and in the other data set you have the fatty acid composition. So, but if you want to compare, integrate those two data sets, you have to convert the second one to the bulk composition as well. Yeah, otherwise you cannot have this direct comparison. And the tool which I like the most in the Lipid Links is a linker, which allows you automatic compilation of all different resources out there for all your hundreds of lipids at once. And I will show you some example of that if you need. So what converter and equalizers are doing. So again, here is an example of PE lipid, plasmalogen PE lipid. And this is a real case scenario, how this exactly the same lipid would be written in all different softwares. And you could see here that it's actually almost impossible to figure out very fast for hundreds of lipids that are written in those different ways. So in this case, we can perform a conversion into the shorthand notation according to Gerhard Liebisch last publication. And we also use here what we say Lipid Links computationally friendly solution, because probably if you work with a big lipid data sets and compiling the data, you might not like the space. So here we use another system, the same one, but just without space in between. And this conversion process, or this converter of Lipid Links, actually became quite good accepted, and it's also we're connecting with different tools. Of course, we connected with our own tools like Lipid Tiger, but for instance, it's completely integrated also in a Biopen. This is a software from Lipid Maps, so it allows you to map your lipidomics data to the different metabolic pathways. So the linker option allows you to retrieve all the different information which is out there for all for your lipids in your list. Yeah, you can do it one by one. For instance, here is an example of PLPC lipid. You get these different abbreviations and so on, notations, and you can get very fast automatically all identifiers and the links for the different databases, for the lion web ontology identifiers as well, but also CAP ID identifiers and KBRIA and UniProt associated IDs as well. And you can do it, of course, for one lipid. But what is the most nice thing, so it looks a bit scary, but it is not. So you can do it for hundreds of lipids at once. Yeah, so you put this is your input names from your experiments, from the software which you use for the identification. So Lipid Links will convert to the shorthand notation, to the this computationally friendly notation, to Biopan IDs, and then you have all information here. For instance, Lipid Maps IDs with Lipid IDs, KBIs, CAC IDs, Lion Web IDs, and also the reactions IDs from RIA, so UniProt and so on. So you have it all at once, and then you're free to process this in any way you like. Yeah, so go to the other tool, so you don't have to click one by one, lipid by lipid. So Lipid Links will do it for you. So this is really a step towards this kind of integration. So again, Lipid Links is totally free. We still didn't really publish it, but it's available on the PowerSheet, so you can use the details over there. And yeah, so there is quite a number of downloads so far, and also we teamed up here with Lipid Maps and they actually host Lipid Links on their website. You don't have to download it if you don't want to install it on your workstation directly, but you can just use this interface supported by Lipid Maps, and you can use it online. And already by now, 3 different published tools integrated within their workflow, the Lipid Links. So with that, I'm coming pretty much to the end of my talk, and I hope I wanted to show to you these all different solutions which we have in our group in order to actually analyze mass spectrometry-derived lipidomics data. And now we are targeting the direction of data integration, where we put our lipid links in between to help to this data transfer process between raw lipidomics data and different type of resources which can support integration of lipidomics data and also lipidomics data with multi-omics data sets. So in the future, so now we are starting to work on this part in order actually to close this complete line. And finally, we can actually simulate some metabolic changes and go back and confirm our hypothesis and different kind of biomedical, biological applications in this case. So by finishing, I also would like to use a chance and invite you, if you are interested, to join a meeting from EpiLipidNet network, which many of us involved. So this is a EU-based lipidomics and epilipidomics network, so our lipid research-oriented network funded by EU Commission, but everybody is absolutely welcome. All our events are free. So if you're interested, next week we have 2 days event online. So just go to the website, check the program, and if you find some interesting talks, just join in. There is no registration in the program. You will find the Zoom link. And with that, I would like to conclude and, of course, thank all the people who involved in this. And once again, so me is absolutely a #1 person driving all these bioinformatics solutions here. But of course, everybody contributes. And thank you very much for your attention.
Thank you so much, Dr. Fedorova. That was a very good and brilliant overview of your recent work, including lots of things that are just recently uploaded to bioRxiv. I see that we have quite a number of questions. Arpana, would you like to lead the discussion?
Yes, of course. Thank you, Dr. Fedorova, for an excellent talk. It was really good to hear the overviews of those tools. I've been curious about them, so it's perfect timing. There's a question here. Could you elaborate on what modifications typically occur in vivo and what in post-sampling artifacts?
Of course, yeah. So if we talk about lipid oxidation, one of the first questions is how do we know that it's not auto-oxidation during our sample preparation? Yeah, so because lipids are very easily getting oxidized just if you leave them on the bench like that. So of course we have to take a lot of precautions. Yeah, so we use a lot of antioxidants like BHT in every single solution which we use to treat from the tissue, let's say, till the mass spec injection, and also cooling down all the solution, making most of the alterations in ice and so on and so on. Yeah, so you have to be really careful and follow certain workflows in order to avoid this artificial oxidation during your sample preparation.
Great. Following up on that question, besides putting BHT, which you just mentioned, to minimize formation of oxidized lipids upon sample prep, she wonders if there are oxidized lipids that represent those formed in sample prep different than those enzymatically formed in vivo. For example, different in oxygen position.
Of course, they might be different because when you have non-enzymatic oxidation, so auto-oxidation during sample preparation, that would be absolutely position specific, right? So it can form in any, not in any, but in any bicellulic position, let's say. Enzymes would do that more specifically, and there is, for instance, 15 logs or 5 logs, yeah, which would introduce the hydroperoxide group in the C15 or carbon 15 or carbon 5 in arachidonic acid. And the problem is that both carbon 15 and carbon 5 oxidized can also formed by artificial oxidation, and then you cannot distinguish that if you do not control for the sample prep here. Of course, at the end, you also compare your, let's say, control versus treatment, health versus disease, right? Or what is also very nice is to use, let's say, reference material, yeah? So to add some sample which you do not expect to have any physiological effects, yeah, so that should be a low level of endogenous lipid peroxidation, and to use the sample during your whole preparation steps, and then to see, do you form some artificial lipids just there. And so we, for instance, when we did our last study on the lipid peroxidations so oxidized lipids in blood plasma of obese and lean individuals. So we used also this NIST SRM plasma, yeah, which is available as reference material. And so there, for instance, with the whole sample prep, we detected some very small amount of oxidized lipids. So we can say, okay, those ones which we detected in our obese or lean individuals, this is the ones which are physiologically relevant in this case.
Great, thank you for those excellent tips to use both on the bench and when you're looking at the data. There's another question on modification. Among all modification sites for oxidation, are those modifications more related to endogenous conditions, or are there specific modifications related to microbiome effects?
Wow, microbiome effects. I'm afraid I cannot answer this question. I'm not sure. I think it depends from the sample you are studying in terms of microbiome effects. Yeah, I have to think about that really.
I guess it kind of relates back to the previous question, you know, in terms of distinguishing where the modification arises from, whether it's the environment or the sample prep or endogenously.
Yeah, I really didn't think about microbiome effects in this respect. Yeah.
Okay, great. We'll move on. In cells, is it expected to see all oxylipids being cooperated into complex lipids such as heat, HOT, prostaglandins, and so on?
Yeah, so what they say that I think there is not available for many tissues or cells, but the data for the blood plasma, I think for humans and rats, they actually showed that 95% of oxalipins, which are usually measured, are coming from those which are esterified to the complex lipids. And because usually when you analyze oxalipins, you, in most of the cases, you do the deesterification first, and then you measure oxalipins. So 95% of your oxalipins is supposed to come from those connected to the complex lipids. So probably the same thing would be in the cells and tissue, because if you think actually the free fatty acids or free oxalipins, this is probably not the most abundant metabolites, yeah, in the cell anyway.
Great,
so I really believe that most of them are esterified to complex lipids.
Wonderful. There's a question here. Do you have any resources that share the perspective of your group during software implementation process, like design choices, challenges, and strategies, especially from the standpoint that there are not software engineers by training?
Yeah, I'm not sure that we have any dedicated resources on the perspectives. But I just want to say that we actually never planned to be a group involved in development of bioinformatic tools. And actually, we just do them because we need them. And that was a super lucky and happy, I don't know, coincidence that we work with me already together for so many years. And he got the interest in actually developing those tools, probably because he got already tired from manual identification back then, and he completely switched from the wet lab to the programming, all of that. And me personally, I do not program anything, right? So we define concept and work together on what it should be and what we really need from the lipidomics perspective, from the analytical perspective, and how to make the tools actually do what we could do ourselves, but much faster.
Great.
But yeah, that's probably the whole perspective of our tool development. So we do them when we need them, and that
perhaps Parker can connect with your main program developer.
Absolutely.
To get more insight on that. Moving on to the next question, does Lipid Links have REST API to be used as a service? It would be very interesting to be used in our chemical translation service, which is used to exchange identifiers, if you weren't familiar.
Yeah, that would be possible, definitely. And just want to say again that in terms of Lipid Links, as well as with any other tools, we are very much open and actually looking for any kind of collaboration. Yeah, so we cannot do everything of that, and we would be happy to actually team up with the people and build up on the Lipid Links platform more and more connections. Yeah, so we did it just the general ones, but would be super happy to collaborate with any other groups who looking for such kind of connections, translations, and so on. We started to do it with Wiki Pathway, I think now, and yeah, so everybody is very welcome just to contact us, and we can see how we can do that.
Great. People definitely want to know about more tools. There's a question here. Do you have any recommendation of tools that predict what modifications can be for some lipids from lipidomics data? We do not do epilipidome. So they don't specifically work with epilipidome, but is looking for a tool that predicts modification.
Yeah, but there is only one tool which predicts modification. So this is, or at least this oxidized modification, yes, Lpp Tiger, right? But I don't know any other tools. And of course, there are more modifications than oxidation out there, obviously, right? So there is nitration and halogenation, at least to mention, right? And there is, I'm afraid there is so far nothing out there. So then you just have to go back to the literature and think what chemically possible at least. Yeah.
Great. And one of the last questions in the chat here, Dr. Tongshan says, I do like the rank index scoring idea, but also wonder if there are cases that the rank index scoring won't work well for certain lipid classes. If you could comment on that.
Yeah, that's of course dependent, yeah, because this rank index scoring, it's actually provides ranks within certain MOZ range, yeah. So, and then we would define, for instance, in the case of phospholipids, so we usually measure phospholipids in the negative ion mode. And we have in our MS2 spectrum, so we have the N ions of fatty acids, yeah, and then the ranking happens within this range where these ions can occur. If you have an overlap of the, let's say, fragment or structure relevant fragments in terms of the MOZ from the different groups, then you can expect the problem here because it will look in the same, let's say, MOZ range and might rank, misrank some fragments. Yeah, it could cause some difficulties. But in this case, which we have, it's usually not.
Great, thank you. Those are all the questions in the chat. I'd like to open it up. Maybe Oliver, if you have, oh, there's one more question here from Oliver. How does your lipid tool deal with ambiguity in the Msms data? Excellent question.
Which exactly ambiguity do we mean here?
Well,
like,
yeah, there's so many ambiguities, right? There's between different types of plasmalogens, like the OE ones. Then there's of course ambiguity in regiospecificity. isomer, like, bonds, where your bonds are, double bonds. So the problem is that usually we don't have enough information, as you know, right?
Exactly. So the point.
This spectrum is this lipid. It could be often a group of lipids. And then that means also, of course, that one lab might, for example, might name this compound zone, so the other one may be more specific. So how do you integrate that, or how do you decide what is the best way to do it?
Absolutely, excellent question as well. So the best thing is, we try to not to try, just do not over-report, right? So if we use a standard LC-MS application like we use without any UVPD, without chiral chromatography, nothing of this. So regular high throughput lipidomics. The best level we can report lipid is this molecular species level, where we know the lipid class and two fatty acid acyl chains, and we do not know which is SN1, we don't know which is SN2, we don't know where the double bonds in these fatty acids. And this is what we actually from our high throughput lipidomics can measure, and this is the level we reported. So we cannot distinguish anything of more sophisticated features. And now it becomes more and more popular with double bond positions, assignment. And of course, for that, new tools needs to be developed. I guess there are a few already out there which can really use the specific fragments provided there to assign, for instance, the position of double bonds. So we have to just report at the level we measure and accept that we cannot do more at the moment.
And maybe a related question is, when we think about the epilipidome, and you add more and more possibilities how things can change, that easily leads to an exponential explosion of possibilities with lots of overlaps, right? All of a sudden you have lots of some formulas that overlap, maybe even spectra that might overlap. So there's, a risk, of course, that all of a sudden you have so many possibilities that you don't know anymore, or it's very hard to rank what is the more likely one, if you see what I mean.
Exactly. This is absolutely what is the problem with oxidized lipids. I showed you this one slide with just calculation how many can be there, and it's an enormous amount. So we do not have an ultimate solution for that, but what we do here is on different levels. So first of all, you cannot have all oxidized lipids possible in one biological system, because the native lipids, they serve as a substrate for oxidized lipids. That means the native lipidome will actually dictate the composition of oxidized lipidome. And as all we know here, that lipidomes of different biological systems are very different. So you cannot, so the lipidome of blood plasma is very different than lipidome of adipose tissue or some other tissue. The first level of actually to shrink the search space is that we use a natural lipidome from this particular biological system to predict the oxidized lipidome. So first we measure natural lipidome in the system in question, predict from this one oxidized lipidome. That already allows to minimize the search space. Then we come to the measurements. So terrible amount of isomers, a lot. So what we can do here is, of course, chromatography. So we now use mostly C30 reverse chromatography, which gives a little bit better separation. The nice thing with oxidized lipids, of course, they are more polar than unoxidized, so they separate nicely, so they look earlier from them. And then we also optimize chromatographic separation in order to resolve isomers. So we know that, for instance, we have hydroperoxide or dihydroxide, for instance, so completely isomeric, the same elemental composition, but dihydroxy will elute after before hydroperoxy. So we checked it a lot with different characterized standard mixtures and so on. So we very much rely here on the retention times as well. And also the thing which we should not underestimate is in-source fragmentation, which is actually happens not in source, but between the atmospheric pressure and the deep vacuum of your mass analyzer. Yeah, so the lipids are very labile, and oxidized lipids are even more labile. For instance, hydroperoxide especially. Yeah, so that means again, without the retention time, it's very easy to get a false positive identifications as well here, and so on and so on. So there are this multi-layers of increasing our confidence in the identification. So in the best case scenario, what we can get now with oxidized lipids, if from the in vivo samples, that we can say, okay, which lipid is that, what kind of modification we have, and in which position this modification is localized. And it's not always possible with positions because those lipids are low abundant in the biological systems, but sometimes it's possible to find the fragments which show this particular position.
All right. Yeah. Thank you again for a fascinating talk. And, you know, I totally agree. Often that's also happened with our lab. You know, we developed the tools because we needed them. You know, we wanted to do biology and then we ended up to do informatics just because.
Exactly.
That's absolutely clear. Are there further questions by other people or comments? Anyone is welcome also to unmute as you see fit. Yeah, that's not the case. Well, then thank you again, Arpana, for leading us through the discussion. Thank you so much, Dr. Fedorova. I would love to host you at UC Davis, but the times they are changing. We cannot always do as we wish, but we all hope that we soon beat the pandemic and then have conferences and so on where we can, you know, properly host and properly talk for an extended time.